Okay. First of all, Hassan, tell me that what do you remember from the previous lesson that we like discussed? What do you remember? What did we study?
So the main part was that the cells are divided in two categories, eukaryotic and prokaryotic.
Yes, cells are divided into two categories, eukaryotic and prokaryotic. Okay. And what were the main differences between these two categories?
So eukaryotic was like plants and human cells, and prokaryotic
Mhm.
was bacteria cells.
Prokaryotic were the bacterial cells. Okay. Yeah, that's good. So we can establish a point over here that prokaryotic are always the bacterial cells. Okay, well done. Very good. And the eukaryotic are plants and animals. But another difference I need to know is that what are the basic differences between the eukaryotic and prokaryotic cells? Like, obviously, you would find the eukaryotic cell in animals and plants, and you would find the prokaryotic cells only in bacteria. But there must be some sort of very clear differences between these two types of cells. Obviously, these two types of cells they are showing some different properties. That is why we divided them.
So
Like,
is it
w-
that prokaryotic cells have only one cell, unicellular?
mhm, the eukaryotes can also be unicellular. Like you may know about the amoeba.
Yeah,
Amoeba is a unicellular organism.
yeah.
So
So isn't that
unicellular
a
eukaryote. No, no, no.
okay, it's a eukaryotic.
Amoeba is a unicellular eukaryote.
Okay.
So one category that is common, you did very good by naming that. This thing is common between eukaryotes and prokaryotes. That eukaryotes and prokaryotes both can be unicellular, but only eukaryotes can be multicellular. But what I'm trying to ask you is that let's suppose I told you that in eukaryotic cells you can find the organelles surrounded by
Cell wall and
organelles
cell.
surrounded by membranes.
Oh, okay,
What does
membranes.
it mean? It means that every organelle has its own membrane. See, it does not mean that it does not mean cell membrane. It means that every organelle has its own membrane. Its own membrane. I can try finding the previous Jamboard to explain this thing, but I would lead you to that Jamboard later. First, try understanding that what is the meaning of membrane bounded ones. Suppose this is a plant cell. This is a plant cell, and in this plant cell, you are looking at the cell wall. And I told you a very important identification of cell wall previously, if you remember it. You asked me that, sir, is the inner one a cell membrane? And I explained a point to you. Do you remember that
So
point?
that gap between the two lines is the cell wall because the cell wall is very thick.
Well done, well done. Cell wall is very thick and cell membrane is very much thin. That is why we often say that the cell membrane cannot be seen under these two layers because the cell wall is too much thick. Okay, so it means that there is a cell membrane underneath this particular two wall structure. Let me draw a dotted line to uh, mention the cell membrane. Suppose the dotted line is the cell membrane. Now, there is no doubt that the cell membrane is there, but it does not mean that when you would look inside this cell, you would not find other membranes. Now, this is a vacuole. This is a vacuole, and once you see at that vacuole, What is the meaning of the oval shape? The meaning of the oval shape is that the vacuole is separated from the rest of the cell with the help of a membrane. The
So
vacuole is separate.
all the organelles inside the plant cell or like inside
Mhm.
the eukaryotic cells have membranes.
Yes,
So even
have
like the
membranes.
nucleus, does the nucleus have a membrane?
Nucleus is has a very fine membrane around it, which is very particular in its working. Vacuum. Let me name a number of membrane organelles which are bounded by membranes, membrane bounded organelles, membrane bounded organelles. So they are further separated by the membrane. Number one that you named, nucleus. The mitochondria. The mitochondria is present in both animal and plant cells.
Cytoplasm?
Then cytoplasm is something that you can say it is providing the ground for all these organelles. Like
Okay.
if I say that cell is a house is built on the ground, right?
Yeah.
So it means that if a house is built on the ground, then the ground for cytoplasm is the ground for the cell is the cytoplasm.
Okay.
So
So
suppose that new. Mhm, please.
they're just like basically it's the groundworks for the whole cell or for at
Yes.
least the organelles.
For the whole cell, for the whole
Okay.
cell, because saying that it works for the whole cell would be better, because there are some functions of the cytoplasm. I tell you later today that that are not done inside the organelles. Only the cytoplasm does those functions. No
Okay.
organelles can do that, which typically means that the cytoplasm has some of its own functions other than supporting the organelles.
Okay. So
So
uh,
nucleus, please.
so then moving on, you could say ribosomes have cell membrane. I mean, have just membranes.
No, ribosomes is one of those organelles which is not surrounded by a membrane.
Okay.
That is why I was enlisting some of the organelles which would help you understand that there are some that are bound by the membranes, but still there are others which are not bound by the membranes.
Okay.
So let's find out nucleus, mitochondria, vacuole, chloroplast, and the Golgi. There is another organelle, very important organelle, Golgi. So all these are membrane bounded. And if another name comes, I would tell you about that later. Okay, then next is next one is non-membrane bounded organelle. Non-membrane bounded. So since you are saying that they are non-membrane bounded, so one of the most important thing about non-membrane bounded organelle is that uh, they are common. No, okay, leave it. The examples of non-membrane bounded organelles are uh, ribosomes. Ribosomes. These are these are basically the important, most important organelle for every cell. Do you know about the function of ribosome?
No.
I'll tell you that later. Ribosomes, without the ribosomes, you cannot even imagine that a cell would work. So
Okay.
it is that important. It is that important. What is the function? We'll discuss that later. So overall, we have a very clear understanding that why we are separating the eukaryotes from the prokaryotes. The very basic thing is that when you would see a prokaryotic cell, now let me take you to the previous Jamboard that we made. Uh, this is the Jamboard that I was explaining previously. Okay, now look at this. If you want the link for this as well, although only two sheets are covered, but we can use that later. So I can change the privacy for this to anyone with the link. and sending you the link for it so that you can access it anytime. Save it with number 2, okay?
Okay. Like when you say save it with number one, like do you mean like download it?
No, 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 no. By this, I mean that make a text document, paste
Okay.
the link, write the number 1 there, and paste the link afterwards.
Oh, okay. I'll do that.
So now see that just look at the difference between the prokaryotic and eukaryotic cell over here. When you would see it, you are clearly looking at some organelles which are clearly clearly expressed, which are clearly bound by the membranes, which are clearly bound by the some boundary. So this is clearly a eukaryotic cell. This one is clearly a eukaryotic cell, and you can see it over here that it is bound. It has very large structures, but all of these have a particular membrane except the ribosomes. Now, ribosomes are those which you can easily see over here. It is common between a prokaryotic cell and eukaryotic. This is a prokaryotic. It is common between a prokaryotic and eukaryotic cell. What other things you can find common? Cell wall is common, cell membrane is common, cytoplasm is common, ribosomes are common. So all these structures are common. Uh, that means that we can find some common features between the prokaryotic cell and the eukaryotic cells. But other than that, you can very clearly see that the eukaryotic cells can be easily differentiated uh, because of the different parts of that are bound by the membranes. So
So,
you can
so
easily
for
see.
for
Mhm.
example, could any eukaryotic cell have DNA or plasmid, or is that unique to eukaryotic? I mean,
It is
prokaryotic.
yes, it is unique to prokaryotic. At this stage, I would answer you that it is unique to prokaryotic. Why am I saying at this stage? Because the every class has some limited knowledge. That is why I'm saying that at this stage we can say that only prokaryotes have plasmid.
Okay.
So you can mention this as a difference between the prokaryotes and eukaryotes, that plasmids are only found in the prokaryotes. Another thing, by just looking at the picture, it seems that these are some membranes. No, 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 no. If I draw it again, I would draw it like this, like a bit rough structure. Now they appear more like a DNA. So basically, they are the DNAs which can be found only in the prokaryotic cells. Okay, so a very clear difference we are finding out. This is one of those differences. This is one of those differences that would help you in all the biology concepts. Always remember, everything that we will study in biology from henceforth, always remember that in biology, everything is done by the cell. As I told you in the first lesson, everything is done by the cell. No doubt, we don't often say that this is done by the cells, but it is a fact. It is a fact that if a heart is beating, if a heart is beating, still it is done by the bunch of cells present in it. So always remember that we need to we need to focus on biology with respect to the working of cells. If we don't take into account that if it's all the cells working, life is overall the working of cells, then it means it would be difficult to understand that what is happening and what is not.
Okay.
Heart is beating, photosynthesis is occurring, I am breathing, my body is running the blood in it, my body is defending me from pathogens, I am working to digest my food. Every single act is done by the cells. So that is why I am focusing on the differences. We are every cell of human being, every cell of human being or every cell of animals is a eukaryotic cell. So we are eukaryotic organisms and the others are prokaryotic organisms. All right. So let's say the eukaryotic organisms contain, and yes, one more thing, I'm sorry, I have to take you back to this slide. One more thing, you can find that the prokaryotic organisms have different organelles, and in about those different organelles, excuse me. Okay, about those different organelles, I have explained a bit of function. Suppose I explained that plasmids are tiny DNAs and they basically contribute to survival of bacteria.
Okay.
They contribute to survival of bacteria, especially when we say that they have antibiotic resistance. Antibiotic resistance. You know that this is something of very much concern. Right now, the pharmaceutical industries are very much about, very much, you can say, worried. They are very much worried about the fact that the bacteria are very quickly getting antibiotic resistant, very quickly. And why is that? Because their plasmids are modifying day by day. They are modifying day by day, and they are getting resistant to every new antibiotic that you are making. So
Okay.
that is a bit concern right now. Secondly, flagellum, I told you about the motility. It make the motility possible. If you remember, motility is there are two types
Helps
of
it move,
terminologies.
basically.
Yes, yes, yes. So that is the flagellum provides. About the rest of the organelles, we can discuss because if we say what is the function of ribosome, we can also understand it by studying the eukaryotes because the function of ribosomes in prokaryotes and eukaryotes are same. Functions are same. That is why if we study the eukaryotes, we can understand the function of ribosomes. If we study the cytoplasm, we can understand the function of the cytoplasm by eukaryotes. When we study the cell membrane, we can understand the function. So the common functions are same. So we can just understand the functions later. First of all, I would like you to have a look at some 3D models I'm going to show you. Now, what do you see? Is it a prokaryotic or eukaryotic cell?
This is a eukaryotic cell
And what make you say that it's a eukaryotic cell?
because it's surrounded by a membrane.
What organelle can you identify that is surrounded by a membrane?
Isn't it like there's one main cell wall as well,
No, no, no,
and
no. Because this you can also find the cell wall in the prokaryotes.
oh, so the, for example, the number, um, I'm not sure.
Hmm, okay, no problem. Um, let's see. Do you do you see a nucleus here? Okay, now hold on. Let me
It's
tell you. Okay, where is the cytoplasm? Ignore the first question. Where is the cytoplasm?
number four, the one, the Light
Yeah, well
green
done.
substance on the floor.
Yeah, yeah, exactly. Do you see that on the cytoplasm there are compartments?
No.
Cytoplasm has compartments. Like number 5 is a separate compartment because it's been walled. The cytoplasm is totally separated from number 5, isn't it?
Yeah.
Focus on number 8. There are multiple organelles like number 8 in the in this
It's like
model.
fixed inside the cytoplasm.
It looks like it is fixed and it is separated. It has its own wall around it. And once you see that inside cytoplasm, there are objects that are separating themselves from the cytoplasm, then definitely it means that the they are organelles. They are membrane bounded organelles. Okay, focus on number 6. It has curved walls, but yet it is surrounded by the wall. It means it is keeping itself separated from the
Yeah,
rest of the
it's
cytoplasm.
surrounded by one connected wall, even though it's curving, it's still one same.
Mm-hmm.
It's the same.
Yes, 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 exactly, exactly. So, what we are looking at a particular cell in which every compartment is separating itself from the main cytoplasm. So, the separation, the compartmentalization, if I tell you properly, over here, the compartmentalization mentalization is only done in the eukaryotic cells. So you can easily see that these all are the organelles. Okay, if I hover my mouse on number 1, what does it say? It says the nucleus.
Nucleus.
If I hover my mouse on number 9, it says the mitochondrion. If I hover on 5, it says the
Vacuole.
vacuole. Hmm. So you see, okay, what make you think? All right, no, let's just ignore the question. Now you see, this is a particular cell that you are looking at, which basically is making you understand that it's a plant cell. Why? One, it contains a vacuole. Second, it contains a
Chloroplast.
particular organelle, chloroplast. Well done. So, yet if you don't say that it's a plant cell, you can just say it's a eukaryotic cell
Okay.
because it has organelles. Now, you got the idea of organelles?
Yes.
All right. Let's see. Let's try and see the prokaryotic cell. A prokaryotic cell. How does it look like? All right. So this is a prokaryotic cell. All right. Do you see any compartments like the previous cell?
No.
We don't see. What's the blue thing? The blue thing that is covering the whole cell. What is that?
Is it a cytoplasm?
Yes, it's the cytoplasm. And what's that entangled thing inside?
Mm, DNA.
DNA. Do you see DNA surrounded by a wall?
No.
You
It
don't
looks like
see.
it's floating. It's floating on the cytoplasm.
Yes, 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 yes. You are 100% right. It is floating. So what does this mean? It is not surrounded. If it is not surrounded, it definitely means that it is a prokaryotic cell. It is a prokaryotic cell. So now we can easily understand that what is meant by the prokaryotic and eukaryotic cell, and always um, other things what we can find. Okay, let's zoom it. Oh, it contains many red spots there. What do you suppose
Plasmids?
are those? Nope.
Plasmids? Oh,
These red spots are ribosomes. Small red things are ribosomes. Those all. Plasmids, about the plasmids, you can see a very transparent thing inside the cytoplasm. This one.
yeah.
Maybe this is representing the plasmid, or maybe this does not contain the plasmid at all, because not all prokaryotes contain the plasmid.
Okay.
But yes, you can now see the difference. You can now feel the difference. Just look at this particular, look at this picture where everything seems to be very organized. Everything seems to be separated by a certain particular uh, wall. And if you see at this model, nothing is separated. So this is one of the biggest difference between these two. And yes, if I take you back to the previous lesson, I told you that eukaryotic cells are almost 50 times larger than the prokaryotic cells. So you can easily estimate that what do they basically, what would they look like in comparison to each other. There is a size difference as well. Now, what's the basic concern over here? The basic concern is that when we look at the diagrams in the book, how would we able to identify, how would we able to differentiate between the prokaryotic and the eukaryotic cell? Like, are you able to would you be able to identify if you see the picture on the book? Because these kind of pictures would not be, these kind of model pictures would not be available on the in the book.
Yeah.
These kinds of models would not be available. So understand to understand this particular um, to understand this particular difference between these kind of cells, I would like you to look at the, hold on, look at the picture I'm just about to open, because identification is very important. Sometimes we get deceived by the pictures. All right. Uh, okay, no, this is the worksheet. Hmm. All right, cell structure. Yes. Hold on. Okay, I guess this is the book which I was looking for. Yes. Let me open the chapter. Chapter is number 18. Page number 24. Okay, this is the cells. All right. So this is just a random animal cell. This is just a random animal cell. I will tell you the differences between animal and plant cells. But right now, you would see some cells which don't look like the animals and plant cells. And where would you find that? You would find it with a particular identification that I will tell you now. Let's first discuss the animals and plant cells so that we can make the things very clear. See, we have established a point that the eukaryotic cells, there are two types of eukaryotic cells. One are animal cells and the other are plant cells. Okay, all right, I have a question. If two things are same, if two things are named same, does that mean they would have a difference between each other or not?
Yeah, they would have a difference. It depends because eukaryotic is a category, and then there could be differences inside the category.
Hmm, well, then you have made things easier for me. Remember that when we say that eukaryotic is a category, then the plant and animal, both cells are you can say eukaryote. But why did we name them differently? If both of them are eukaryotes, why did we name them differently, animal and plant? It
Bec-
means that no doubt that being eukaryotes, they are same, but there must be a very particular difference between them due to which we are naming them differently. Like a phone, a mobile phone called as iPhone. Okay. What difference do you find between iPhone 3 and iPhone 13? Obviously, both are iPhone. You would name both as iPhones.
Yeah,
Both the two people, one that has iPhone 3 would say that I have iPhone. One that would have iPhone 13 would have would say that I have iPhone. Both are saying the correct thing, but obviously there are differences. Now, in that sense, we can say that both are eukaryotes, but they have differences between each other. Okay, then what differences we can find? In eukaryotic organisms, in the eukaryotic organisms, the animal cells, first of all, the one thing that you would find in an animal cell is the shape of it. Or every time you would draw a plant cell, the, every plant would have almost a very definite shape. It would always have a very definite shape. Like it can have a shape like rectangular, or maybe octagonal, or maybe pentagonal. Just look at this picture. It is a pentagonal picture, but its shape is defined. Shape is always defined. So if you see a cell that has specifically a circular shape, because at your level, when you discuss the animal and plant cells, every circular shaped cell is animal cell, circular shaped cell.
yeah,
And every rectangular or a defined shaped cells is a plant cell. So first difference is the shape.
okay,
All right, fine. The first difference. Second, second is you would never find anything like a double layer inside an animal cell. This would only be found in plant cell. And what does this mean? This means cell wall. You would never find in animal cell. I was just referring to this one. If you color this hole, this is the cell wall.
Yeah.
You would never find it in animal cells. Okay, let's discuss a situation. The situation is, what organisms contain the cell wall, or what organisms do not contain the cell wall? Both are questions of same category. Okay, let's discuss what organisms do contain the cell wall.
So all plant organisms like leaf cells,
Mhm.
um, anything that is not an animal cell and is a plant cell has a cell wall.
Anything that is not an animal cell, does it mean that fungi also possess cell wall?
No, I mean like anything that is not an animal cell but is a plant cell has a cell wall.
First section of your sentence is right, but second section is wrong. Remember, every organism except animal would have a cell wall.
Okay.
Suppose fungi, yeast, they contain a cell wall. And just in the previous lesson, we discussed that the prokaryotes also have a cell wall. Similarly, talking about algae, another type of particular organism that can perform the photosynthesis, they also have a cell wall. So you cannot deny the fact that the animal cells are only cells that would not contain a cell wall. Okay, next. Next thing that you would find in an animal cell is that the largest organelle in an animal cell is in the middle of it, and that is the nucleus. But nucleus is not the largest organelle in the plant cell. Instead, the largest organelle in the plant cell is the vacuole that you would not find in the animal cell. You would not find it in the animal cell. Got that?
Yeah.
Third, okay, another organelle you would find in both animal and plant cell, and that is
Mitochondria.
the mitochondria. So you would find these organelles in both of the plant cells. Okay, then yes, you would also find the ribosomes very much closer to the nucleus. You would find them. So the ribosomes are also common. Let me label them: mitochondria, then ribosomes. What you do not find is the chloroplast, an oval shaped organelle which has some basic working related to the plant cell. Related to the plant cell. And okay, now uh, the cell wall is the difference. The four differences: you should complete. One is shape, second is cell wall, third is vacuole, and fourth is the chloroplast. Chloroplast. Now, obviously, we need to discuss about the differences about different organelles, that what are their functions. But most importantly, the vacuoles cannot be found in the animal cell. The vacuoles cannot be found in the animal cells, or you can rarely find the vacuoles. You can find the vacuole, but very rarely. All right. Now, if you see, first of all, if you see an image of a plant cell or an animal cell, first of all, how would you identify?
Here this is an animal cell because of the cell membrane. So we
Hmm.
know that it is, first of all, that it's a eukaryotic and it's not a
Hmm.
prokaryotic.
Hmm.
And wait, so it has a vacuole. So I thought vacuoles are only for plant cells, or can they be in?
I said a few seconds ago that they are rarely found in the animal cells, but this is a big mistake. I do not think that they should have mentioned it over here, because they are very rarely found in the animals, the vacuoles. So I would suggest you to ignore that thing. Okay, about the rest of it, the very first identification of an
Shape.
animal cell is the shape.
Yeah, the shape.
Yes. So the shape, you see that this shape is not a regular shape. Instead, it is not even a proper circle. The boundaries are not smooth.
Yeah.
The boundaries are not smooth. They are just like you can say rough boundaries. And that is because it's not a cell wall. Cell wall makes the boundary very smooth. Secondly, at this level, you can say that this particular cell uh, this particular cell do not show the vacuoles, because I said just ignore the fact that they are showing the vacuole over here. Third, the very important thing, you would find the largest organelle in the animal cell, and that is the nucleus. But if we talk about the plant cell, that is not the nucleus. So this is another difference. The difference of the largest organelle. In plants, it is the vacuole, and in animals, it is the nucleus. Another difference, by just looking at the picture, you can see that you cannot find any chloroplast. You cannot find any chloroplast. Now let's have a look at the plant cell. Okay, you can see easily that the vacuole or vacuoles are there. Second, the chloroplast can be found. Third, very smooth and thick outer surface. See, this is another way of understanding the picture. When you see the outer surface, it is very thin in the animal cell, but in a plant cell, it is very thick. Proper uh, double, it seems that there's a proper double membrane around it. You can see the chloroplast, and you can see another very unique thing which is not mentioned yet. I have not mentioned yet, and that is the starch grains inside the chloroplast. So that is another difference.
Okay.
So these are a few differences with the help of which you can easily identify or differentiate between the plants and animal cells. Right now, I'm about to mention the differences. The shape is regular in plant. This is for plant cells. And in animal cells, its
Irregular.
shape is irregular. Cell wall present,
Not present. Animal.
not present, or absent. Vacuole one large,
An animal is rarely present.
absent or rarely very small. They don't have very large vacuole if it is present. Chloroplast is present,
Absent.
absent. Largest organelle, vacuole,
Nucleus.
nucleus. So you can easily identify the differences, and you can also identify it just looking at the picture. So it's very easy to understand. Any problem till here?
No.
Okay. So now, since we have finalized the things, let's draw the rest of the structures which are common. Cytoplasm is common, and then cell membrane. Now, if you look carefully in plant, I have not uh, marked the cell membrane. I have marked the cell membrane just below the second layer, just below the second layer, because
Yeah.
the cell membrane would be found there, not at the second layer. So that is the reason I marked it there. Okay, the cell membrane. Now let's start talking about the functions of different organelles so that we can understand what a cell performs. See, now we not for the not for the sake of the cell. Remember, we would now try to understand about the organelles for the sake of the functions of the body. Like obviously, every cell is working for its own safety, for its own survival. Every cell. But it is a reality that if you observe, all the cells basically are working for the body when it comes to collective working. So the very first organelle is called as a cell membrane. It is um, outermost in animal cells, and it is, like you can say, present inside the cell wall in plant cells. Inside the cell wall in plant cells. Third feature is that it is living. It is a living organelle. Now, I would discuss the living and non-living organelles a bit later. And what does it do? It controls the transmembrane transport, or you can also say it controls the movement of particles, movement of molecules, ions, or particles inside and outside the cell. Outside the cell. So this is very important to mention that it controls the movement. All right. So cell membrane, it is uh, it is also called as partially, differentially, partially permeable or differentially permeable Or a third word that we can use is selectively. Selectively permeable membrane. And why is that? Because it selectively allows the particles to move. Selectively allows the particles to cross it. That is why it is mentioned as the selectively or partially permeable membrane. Got the idea?
Yes.
Okay. Now, I would tell you about why am I saying that it is a living organelle after discussing the cell wall. Cell wall absent in animals, absent in animals. It is outermost boundary of plant cells. Another thing is it is very rigid, very rigid or hard. In plants, it is made of cellulose. It prevents the plants from the pressure exerted by water from inside. Exerted by water from inside. Or this pressure is called as turgor pressure. Okay, all right, another thing, cellulose is a chemical or molecule. All right, now, one thing that is quite important to understand, and that is, suppose that we are like we say that it is outermost boundary. Yes, another thing, it never controls the movements, never controls the movement of particles crossing it, or even does not have any other function, any other function. Its only role is act as a wall, is to act as a wall. So overall, you can say the cell wall is considered as a dead organelle. It is considered as a dead organelle. Now,
Because it doesn't have a like active purpose.
yes, that is why it is considered dead. So you can now justify that why the cell membrane is considered as living wall while the cell wall is not considered as a living structure. Cell
Because
wall is dead.
because the cell membrane is actively allowing or preventing particles from going in and out the cell.
Yes, yes, that is the reason. So overall, you can just simply say that they both are, although the layers of the cell, the outer parts of the cells, yet you would not treat them equally. Okay, secondly, let me tell one more thing about the cell membrane. It is made of molecules, the proteins, lipids, and carbohydrates. Three types of molecules which are found in living organisms. So it means that they also have a chemical difference because the cell wall is made of cellulose. Okay, one thing I would like you to know, very important, yet it would serve an important purpose to you in understanding of the organelles, and that is all organelles, all organelles are made of molecules. It is the molecules that would combine to build up an organelle. So it is important to understand that every organelle has the molecules as the cements and bricks.
Yeah.
All right. Now let's talk about the next organelle, that is the cytoplasm. Okay. One function of the cytoplasm is to act as the ground material, right? Ground material for the cell. It provides space for the organelles and site for metabolic reactions. Metabolic reactions. Another thing, it stores wastes and food. Stores wastes and foods, and all of them have the same purpose. All of them have the same purpose. What do I mean by all of them? By that I mean uh, whether it is found in the prokaryotic cell or eukaryotic cell, all have the same purpose. So that is the important function of the cytoplasm. Other than that, you don't need to know any more function of it. Okay, next organelle could be the nucleus. Or okay, let's discuss the nucleus first. Nucleus, as I have told you, that it is found on the side in the plant cells, while in animals, while central in animal cells. And uh, it contains a material called as nucleoplasm. Now, this is important to understand that the cytoplasm is a separate thing and the nucleoplasm is the separate thing. The cytoplasm is outside the nucleus. Nucleoplasm is inside the nuclear membrane. Contains a material called as nucleoplasm, surrounded by a membrane surrounded by a membrane called as nuclear membrane or nuclear envelope. Uh, contains pores, contains small pores called as nuclear pores. Nuclear pores. Another thing you will find out about the nucleus is that it contains a material called as chromatin material. Chromatin material basically is the name of thread-like DNA molecules. Thread-like DNA molecules clustered inside the nucleoplasm. Clustered inside the nucleoplasm. Another thing about it is uh, it also contains a thick, a thick circular region, circular central region called as nucleolus. Called as nucleolus. So that is what how the nucleus appears. Okay, first I would try drawing the nucleus by myself. Suppose that this is the nucleus possessing small pores. Nucleus, this is the nuclear envelope, and these are the nuclear pores. This is the nuclear envelope. These are, this is the chromatin material. Which basically is the cluster of DNA molecules, chromatin material, and in the center of it you would find a very thick body, which is what? Which is the nucleolus. And this is the nucleoplasm. This is the nucleoplasm. So it may appear like this, but I can show you a model of it so you can understand it better. Just look at the center. Okay, first of all, tell me what kind of cell it is. Is it a plant cell or an animal cell?
Which cell?
This one.
Oh, so it is a eukaryotic cell.
Mhm.
It's an animal cell
Okay, what made you say that?
because I can see the nucleus, but I can't see the vacuole, and the nucleus is the
Good,
biggest organelle.
good, good, good. This is a good identification. Yet, you cannot find the chloroplast as well, right?
Yeah, but the plant cell doesn't necessarily have to have the chloroplast, right? Because
That's
that's only
also
the
a
leaf
very good
cell.
assumption. Very good assumption. Very good. And we cannot find the vacuole. That's a very good assumption. And secondly, although you may see that it's a regular shape, but if you look at this from this angle, it does not seem to
Yeah,
have a regular shape.
yeah,
All right, now let's have a look here. This is the nucleus, and you can
yeah.
find these small gray things. These gray things are nuclear pores, and the outer wall obviously is the nuclear envelope. And in the center, you find a dark region. The light purple things, these are the DNA strands. And in the center, you find the darker region is called the nucleolus.
Okay.
It's called the nucleolus. And if you want to find it out with the microscopic picture, you can see. Now, how would we identify with this picture that it's a nucleus? First of all, you are going to look for the central region. Does it contain the nucleolus? Yes, it does. Secondly, uh, you can look for the membrane, nuclear membrane, although it's not a very clear picture. But if you find the central thick region within a boundary, it definitely is the nucleus. It definitely is the nucleus. So that is how you identify. Although these kind of pictures don't appear in the paper. These kinds of picture don't appear. These appear in the A levels, but not in O levels. Or in IGCSE maybe. So
Okay.
you got it?
Yes.
Okay, our time is up. One very important thing I need you to know, very, very much important, and that is always remember that our cells are drawn in a 2D shape. When I was showing you the pictures from the book,
Yeah.
all those pictures are in 2D shape.
Yes.
But in reality, every cell is in 3D shape. See, suppose this is a structure. My
Yeah.
hand, this is a structure. But and these are all the side of the structure. When it comes to drawing it, if you draw only this side on the book, what about the previous? What about the side that is on the back?
Yeah.
That would totally be misunderstood. It means always consider that every cell is a 3D shape. It's a 3D model. So it means the pictures that you look at, they actually are drawings made after looking at the cells under the microscope. Now, the microscopes normally have the vision of 2 dimensions. Normally, the microscopes that we used to use 10 years ago, yes, not more than 10 years ago, those were light microscopes, and light microscopes don't show you a 3D shape of a cell. They don't show you. The actual 3D shape of a cell can only be seen in the electron microscope. So every structure, whether it is nucleus, whether it is chloroplast, mitochondria, or even a whole cell, it's 3D. You know what is the shape of the cell? We often say the cell is circular. No, the cell is spherical.
Yeah.
Spherical mean like a tennis ball.
Yeah.
If you cut the tennis ball from in between, it does not mean that other part was not existing. The
Yeah.
tennis ball was an intact structure. You cut it from the middle. If you cut it from the middle to study what is inside, that is only one side. The other side is still there. It means you have to consider every cell, or a cell like a tennis ball, a 3D shape. Got it?
Yes.
So I will tell you this tomorrow again, and then we'll see the diagrams with this perspective. And we'll come to understand that why it is very important to understand that every cell is a 3D shape, not a 2D shape.
Okay.
All right? Okay then. Thank you very much.
हमारी अगली क्लास थर्सडे को होगी.
Yes.
ओके.